Hello, my name is Erica Davis. I'm a human genetics researcher and have been working to understand the causes of BBS for over 15 years. My laboratory is based at Lurie Children's Hospital of Chicago in affiliation with Northwestern University. The goal for this video is to give an overview of some of the approaches that researchers use to study BBS genes and proteins. BBS is a genetic condition, which means that it is inherited in families. So how are causal BBS genes identified? We are living in a truly fascinating time in medical genetics. We know that the human genome is made up of about 22,000 genes, and that there are at least 27 of those that can cause BBS. Technological advances have reduced the time required to complete a clinical genetic test for BBS, and have also reduced the cost. So if a person has several of the symptoms known to be involved in BBS, a doctor might order a diagnostic test. Now, one example is something called a panel sequencing test. Sequencing is reading of the DNA letters that make up a gene, and a panel means that a subset of genes in the human genome are read. This subset includes the known BBS genes. Now, another example is a whole exome sequencing test. This means that the DNA letters of all 22,000 genes of the genome are read. Now, regardless of the type of test, once the DNA letters are read by a machine called a sequencer, the laboratory will search for DNA changes in the BBS genes that fulfill 3 criteria. So first, the DNA changes need to be rare in the general population. And this is because BBS is a rare condition, so we would not expect causal DNA changes to be common in generally healthy people. Second, the DNA changes should be inherited from parents of the person with BBS. And third, the DNA changes are likely to affect the way the BBS proteins function in the cells of the body. Now remember, genes are like instructions or a blueprint, and the proteins are the actual building blocks that make up the cells of the body. Now, about 80% of people who likely have BBS will receive a BBS gene diagnosis from a clinical genetic test. For the 20% of people whose tests are inconclusive, they can participate in genetics research studies that aim to discover new BBS genes. We know from nearly 20 years worth of research that BBS proteins are important for making cilia and allowing them to function properly. How do you study cilia? Cilia are fascinating and important compartments on nearly every cell type of the body, and there are many ways that we can both visualize the structure of cilia and also test whether cilia are functioning properly. Now, much of this work is carried out in cells that grow in a dish. Cells are the basic unit of life that make up organs and cells of the body. Researchers sometimes use skin cells from humans, and you or someone that you know may have given a skin punch biopsy for research. Additionally, there are classic laboratory cell lines used in BBS research. These might include kidney cells, retinal cells, or connective tissue cells called fibroblasts. We can answer so many questions about cilia with specialist stains and high-resolution microscopes that enable us to see this tiny cellular structure during normal circumstances and also when a BBS protein doesn't work as it should. For example, it is important to study the length of the cilium and its position on cells. It is also important to know where BBS proteins are found in cells and with which other proteins they interact with. So for example, we know that there is a group of 7 BBS proteins that form a complex known as the BBSome, which moves proteins to and from the primary cilium. So the cilium is comprised of about 1,000 different proteins, and it is also important to know where these proteins are located when a BBS protein does not function properly. Additionally, we know that cilia are like the antenna for the cell and transmit and receive signals from its neighbors. Biochemical experiments allow researchers to understand the type and strength of signal that is sent and received in cellular models of BBS. What are the different types of animals that are used to study BBS proteins? Any animal that has cells that make cilia can be used to study BBS proteins. Most often, vertebrate animals, or any animal with a backbone, are used to study BBS. There are laboratory mice that have been engineered to study nearly all of the known BBS genes. Additionally, there are naturally occurring animal models, such as the BBS2 Shetland sheepdog or BBS8 golden retriever, or there's even a BBS7 monkey. Non-mammalian models, such as zebrafish, have also taught us a lot about BBS. There are also BBS worm, fly, or green algae models. Animals have a lot of similar developmental and degenerative processes that take place in the same way they do in humans. Now, the most predominant feature in most BBS animal models is blindness. Among BBS mammals, obesity is also a common aspect of the syndrome, with greater variability in the way that other organs, such as the kidneys, are affected. However, there are some common symptoms in humans that are rarely observed in BBS animals, such as polydactyly or extra fingers and toes. The use of animals in BBS research is critical for several reasons. They allow us to study the way BBS proteins work within specific organs of the body and during specific times in the course of an animal's early development and lifespan. That can't really be accomplished by studying cells that grow in a dish. Considering that there are so many different types of animal models of BBS, how does a researcher choose which animal model to use? For any animal model study, the same gene humans have need to be present in the genome of that animal. Now, the good news is that all of the BBS proteins discovered thus far can be found in vertebrate species. Most, but not all of the BBS proteins can be found in animal models that do not have a backbone, such as worms or flies. The next important consideration is the similarity of the organ system that you want to study between the model animal species and humans. For example, any animal with a retina would be appropriate to study progression of blindness in BBS. So the mouse or the zebrafish would be a good choice, but the worm might not be as helpful. It is also important to choose an animal model based on numbers needed to answer the experimental question. For instance, a pair of mice can produce 6 to 8 offspring every 21 days. However, a pair of zebrafish adults can generate about 200 offspring every 14 days. So tied to the question of numbers is a question of animal cost, which is not surprisingly related to the size of the animal. Imagine a mouse cage. For a research laboratory, it can house 5 mice at an approximate cost of $1 per day, which means that a single mouse costs around 20 cents per day. Now, imagine a fish tank. For a zebrafish, which are pet store fish that are about just 1 inch long, they live in tanks that cost around 30 cents per tank per day, and you can keep up to 30 fish per tank. This means that the single animal cost is just 1 penny or less per day. Now, often, researchers use multiple animal species to answer questions about BBS. My lab, as well as others around the world, have used small animals to make an initial discovery. Then, researchers can confirm and expand on that discovery in a larger mammalian model, such as a mouse. Who covers the costs to fund BBS research? BBS research advances through the actions of people, places, and things. The people are scientists with varied educational backgrounds and career stages. The places are typically laboratories that may be in an academic setting, such as a university or a hospital, or even in a company. The things required to do the work include laboratory equipment, laboratory animals, and even tubes and special reagents for experiments. Funding is required to maintain this wonderful mix of people, places, and things. Now, BBS research in universities is usually funded by major government funding agencies, such as the National Institutes of Health in the United States, and researchers have to compete every 2 to 5 years for funding. Sometimes, nonprofit organizations, such as the BBS Foundation, fund researchers, but this is less common. This kind of research often costs hundreds of thousands or even millions of dollars over several years, and not many non-governmental organizations can raise that much money. Companies with an interest in BBS may also fund research and development activities. Why is it so important to understand BBS genes and proteins in people and animals? Researchers are motivated to discover new information to help families and their doctors. Information about DNA changes in genes that cause BBS can help decrease the number of tests and reduce the time to achieve a diagnosis. Understanding how BBS proteins work and knowing what goes wrong in cilia and cells when BBS proteins don't work properly is an important basis for developing therapies. Detailed knowledge about ciliary structure, ciliary function, and the ways in which cells talk to each other through cilia are a starting point toward finding a drug or therapy to correct it. Once new therapies are optimized in animals to ensure that they are safe and can improve BBS symptoms, then these therapies can advance for trials in humans.